Hello everyone, my name is Christina and today I will be talking to you about Poultry Disease Diagnostics 101, so finding the right tool for the job. So what are our learning objectives? So first is to identify the common diagnostic tests used for pathogens of chickens. And second is to find out how they're used, so by defining the target of these diagnostic tests. And then the third learning objective is to contextualize this diagnostic test into a clinical or field setting, so placing it on when and where it can be used. And then I would also like to mention here that we'll be focusing mainly on ELISA, which is a serological test, and also PCR, which is a nucleic acid test. And so before we start, I also wanted to mention this prototypical life history of a pathogen and host. And so on the x-axis here is the number of days after an animal is infected. And also we're like to mention that we're using infectious bronchitis virus as an example. There are certainly other pathogens and other lifestyles that we have, but for the sake of discussion, we'll use infectious bronchitis. And here, highlighted here are three events in that life history. So first is viral replication, where we can actually see, or we can actually detect the actual virus and its genetic material. And so we see here that for bronchitis, it's about at 3 or 4 days after an animal is infected, where we see the rate of viral replication very high. At its highest, actually. And then after there's actually a delay where we can see clinical disease, which is the red line, and for bronchitis it's called ciliostasis, where the cilia of the trachea stops beating. And you can see here that there's about a day or two of delay where we can actually see clinical disease. And then way out after about 2 weeks, then when the virus is gone and the clinical disease is gone, then we can actually detect the immune response that is made by the chicken. And so I'd like to here just to examine here that none of these three peaks actually overlap perfectly, and at any given time you may or may not see virus, or you may or may not see clinical disease, depending on where you are in the timeline. Okay. So now let's now go into diagnostics. So what is diagnostics? So diagnostics are activities that aim to identify the presence or absence of disease for the purpose of prevention and management of diseases. And I'd also like to mention here that by diseases we mean diseases caused by pathogens, because there are certainly other diseases that are not caused by pathogens. For example, toxins or management. Okay, so what are the tools that we have in our diagnostic kits? So there are basically 3 kinds of diagnostic kits, of diagnostic tools. First is the isolation and characterization, or the classical method, so actually finding the actual bacteria, virus, or parasites, for example. And then the other 2 are based on molecular, so are molecular based. So basically they're looking at portions of the actual biological agent, and it can be serological based, in other words, based on antibody-antigen interaction, or nucleic acid based, so based on genetic material. And we'll discuss more on that as we go along in the presentation. So first off, so what are the classical methods? So the aka classical method is the isolation and characterization of pathogens itself. So we are actually trying to find the actual virus, bacteria, or parasite itself. And so it really depends, the methods that are used really depends on what the pathogen is. And so this the isolation and characterization of the pathogen itself is definitive. So if you do get a positive for isolation of bronchitis, for example, you definitely have bronchitis, but it's very time and resource intensive. But there is still a space for this diagnostic tool because it's very important for generating type strains and also very important for vaccine development. And so here's just an example of an isolation flow chart for salmonella, and then I got this from the laboratory manual of the avian Association for Avian Pathologists. And you can see here, like, if you have two different types of sample, organ tissues or intestinal environmental samples, they go through all of these steps just to be able to identify whether or not it's positive for salmonella. And it can take between two days to a week to get the results. So it's very labor intensive. It's also time intensive. Okay, so what about the molecular methods? So, as mentioned earlier, the molecular methods detects portions of the pathogen itself, either the proteins or nucleic acid, so which can be RNA or DNA depending on the agent that you have. And so the advantages of the molecular methods are actually quite a number, because there is an increase of the number of samples that you can test. There is an increased speed in testing, so in a matter of hours instead of days. And typically it's more sensitive and cheaper than the classical method of isolating and characterizing a pathogen. So one disadvantage, though, of the molecular methods is that interpretation of the results is less straightforward. So, and we'll discuss more on that, of why it's less straightforward than the isolation characterization of the actual pathogen. So in the molecular methods, we have, again, as I mentioned earlier, the serological and nucleic acid base. So for the serological, we are using the antigen-antibody interaction as a way to detect either the antigen, which is the target, which is usually a protein, or the antibody, so the immune response that is produced when a pathogen infects an animal. And so in the schematic here on your right, the orange circles is the antigen, and the antibodies typically diagrammed as a Y-shaped molecule. And so in our serological methods, the most common method by far is ELISA, or which is short for enzyme-linked immunosorbent assay. So a really long name. But again, so the reason why there's an enzyme in the name is that we actually use an enzyme that produces either color or light to be able to detect the interaction between the antigen and antibody. And so depending on how you design your ELISA, it can either detect the antigen or the antibody. And there's examples in the bottom of the slide. So you typically direct ELISA. So direct ELISA typically detects the antigen. And so when there is antigen present in your sample, the enzyme that is attached to the antibody will produce a color, giving you a positive result. And then on your right is an example of a Sandwich ELISA, which is typically used to detect the antibody. In other words, the immune response against the pathogen. And again, if you have a positive sample, then you will get typically a color, which is produced by the enzyme that is attached to one of the antibodies. Okay, so what about our nucleic acid-based methods? So for this, kinds of methods, we're we're trying to detect the genetic material of the pathogen, which can be RNA or DNA. And so one important thing to mention here is that these methods can detect viable and non-viable pathogens. And so if you have genetic material, it does not mean that the pathogen is viable or not. So there's it may or may not be, just important to note. And then one of the most important reasons why nucleic acid based methods are very robust is that because it takes advantage of the uniqueness of our gene sequences for different pathogens or for different biological agents as well. So for example, if we have let's say if we have two different agents here, we have bronchitis with this, for example, sequence in its genome, and salmonella with the sequence at the bottom with the genome, we can use basically the same chemistry, so the same methods to detect, but we just change the sequence of the primers or the probes to make the test specific either for bronchitis or for salmonella. And so one of the most common nucleic acid based methods is PCR, which is short for polymerase chain reaction. And this test is very sensitive because there's an exponential amplification, so not just geometric, but there's an exponential amplification of genetic material, so it's highly sensitive. So for example, if you have one DNA molecule that you put into your PCR reaction, you can get in 1 or 2 cycles 8 copies of this one single DNA strand. Okay, so now we've discussed of what the diagnostic tests are and how they can be used. So the next step is to talk about when and where these diagnostic tests can be used and where does it fall in the life history of the host and pathogen. So if you recall the first, for earlier slides, we discussed the life history. So where does the diagnostic step fall under the life history of the pathogen? I like to think about that. Okay, so again, we're using infectious bronchitis as an example, and so let's first discuss the first event in that life history, which is the viral replication. So what can we use to detect viral replication? And so first, there are actually quite a number. So we can either isolate the virus, right? Or we can detect this genetic material. Or we can also try to detect the portions of the virus itself. So using an ELISA for the antigen for bronchitis, for example. And again, this would work well between 3 to 5 days into the infection cycle, right? And then so as you know, as poultry practitioners, actually we usually first look at or see that there's a problem in our flock through clinical signs. So actually, if you would notice, there's a single delay, maybe 1 or 2 days between the highest peak of viral replication and the actual clinical disease. You notice here. And then finally, after 2 weeks, so if we use an ELISA to detect for antibodies, so in other words, antibodies that are target bronchitis, that is actually 2 weeks out. So we will probably not be able to use virus isolation or PCR or ELISA for the antigen at this point in the infection cycle. So just to give you an idea of how this all plays out temporally. Okay, so now let's all apply our knowledge. I will give you time to read over this case study, and it's called Old or New. And so we have 3-week-old broilers who have clinical signs: 2 out of 10 with rales, 1 out of 10 with conjunctivitis, 3 out of 10 with erysiculitis. And its vaccination history is a spray vaccination with Newcastle, infectious bronchitis, and coccidia at the day of hatch. And when you need crop seed birds, we did colonial swabs and blood, and 5 out of 10 were positive for ELISA, and 3 out of 10 were positive for PCR. RT-PCR from bronchitis, and the diagnostician told you that it's a vaccine type. And so, what would you recommend for management? So I'll give you time to discuss between you and your seat mates. And so if we have time, there is a second scenario. And so here, if the birds were positive, all the birds that were necropsied were positive for ELISA, and they were unusually high, 10 times normal for vaccinated birds, what would you recommend? Okay. Good job, everyone. So what is our take-home message? So I would like you to think about these 2 questions when you either order or interpret diagnostic tests. So first, find out the what. So what is the target of the diagnostic tests? Is it detecting antibodies? Is it detecting the antigen? Is it detecting material or the pathogen itself? And then second, once you have this diagnostic tests, the results, where does it fall in the life history of the pathogen? So again, depending on what you're detecting, it may be 1 day, it may be 4 days, it might be 3 weeks or 2 weeks into the life history of the pathogen. And that's really important. It's to be able to interpret the results accurately and to be able to respond with management appropriately. And so and always remember to use the right tool for the job. All right, so thank you all for your attention. If you have any questions, please feel free to email me with my email on the screen. And thank you so much.